There's several ways of genetically modifying a fruit fly, but the overall process is that you collect embryos and you have a very, very fine glass capillary needle, and then you insert that into the embryo, and then you inject a very tiny amount of this mixture. And what happens is then the DNA that you inject ends up being incorporated into the DNA of that fruit fly. The reason we chose fruit flies is because they're very easy to genetically manipulate and they have a very fast life cycle. And so the idea is essentially to use the fruit flies as a platform to prototype how this technology would work in mosquitoes or in fish or other types of animals. This is essentially a platform technology that enables a lot of other breakthroughs. And so what we were doing is we were anesthetizing the flies using carbon dioxide. They're in a little vial. And what we do is we put a little puff of carbon dioxide into that vial. And then that causes them to pass out. And then we can open that vial and then we can just dump them on this pad. And then that pad is actually porous and there's carbon dioxide that's coming up from underneath that. And so that is supplying enough carbon dioxide to keep those flies unconscious while we work with them. When we make changes to insects or anything else, we usually have something along with that change that's easy to see. You have something that's called a marker, and that marker is something that lets you see what has been genetically engineered. So what the fluorescent eyes are telling us is that these are flies that have been genetically modified. And so when we see the fluorescent eyes, we're like, those are the flies that also have the mutation that we're most interested in. We can apply this technology to help to either control populations of mosquitoes that spread diseases or to engineer synthetic species in a way so that mosquitoes are no longer spreading diseases. Two ways that we could apply this technology to Aedes aegypti, which are the mosquitoes that are primarily responsible for spreading Zika, dengue, yellow fever, chikungunya. One of those is that we make one of these synthetic species that are incompatible with the wild type, and then we engineer it so that it no longer has the ability to spread those viruses. Our engineered mosquitoes will displace the wild ones and then occupy that ecological niche. You'll still have mosquitoes, they'll still bite, but they'll no longer be able to transmit disease. The other application has to do with suppressing those populations. We can control whether or not females can reach adulthood. Only males will emerge. Those males will mate with wild females, and then those females won't have any offspring, and that'll help to drive the population down. Ecologists that have looked at this have essentially found that for this mosquito species, there isn't anything that's so dependent on it that if we got rid of it, that there would be some kind of negative ecological consequence. That being said, if it turns out that there is a negative consequence, we can always bring them back. This type of technology is going to be much more accessible to areas in the developing world, and it'll help them to control the mosquitoes that are responsible for millions upon millions of infections and hundreds of thousands of deaths every year.